All right, perhaps we'll start now. We're giving the annual Macular Research Group Patient Day. Today, I'll be talking about the laboratory research that we do, and Matt Simunovic will be talking about the way that he approaches macular diseases and some new treatments that group is considering. They do more gene therapy and genetic diseases, but they also have some interesting new potential treatments for macular disease. At the last meeting we had, I talked about some new developments in the clinical trials unit. One of these was Vabysmo, the new VEGF inhibitor for wet macular degeneration, for diabetic macular edema, and for retinal vein occlusion. That actually has been released now, and it's in our practices, and we're using it. And we do seem to be seeing a benefit in some patients who are requiring a lot of injections, and you'd like to give fewer injections on the ranibizumab or the Lucentis and Eylea. Some patients it doesn't seem to make much difference in, but some after 2 or 3 injections, they do seem to do a bit better and they can be treated less frequently. So you might want to talk to your doctor about that. The other one was the pegcetacoplan, which is the first drug which may slow the advancement of advanced dry macular degeneration or macular atrophy. This is something we haven't been able to treat yet. This drug has now been released in the United States. They usually released there, and it's being considered now by the Therapeutic Goods Administration, and we're hopeful that that will be released in the first quarter of next year, around that time. For that drug, a reasonable summary might be for every year of treatment, you might put off the loss of whatever loss of vision that you are destined to experience, you might put it off for, say, 2 months. So after 2 years of treatment, it might be 4 months. After 6 years of treatment, it might be 6 months. It could be something like that. It's not a cure by any means, but it's a step in the right direction, and there's some patients who will definitely benefit. So today I'm talking about what the laboratory research unit does. The Macular Research Group at the Southside Institute has the lab, the basic research unit, and we look at basically macular diseases. And we're looking for new targets to treat macular diseases such as macular degeneration, but also diabetic macular edema is an interest of ours. Retinal vein occlusion is a common disease in the macula, and also MacTel. We're one of the major centres for that. The clinical trials unit, which is what I presented on last year, they do clinical trials in all conditions. At the moment, we still have some trials of for atrophy, which are enrolling, and that's going to be for a genetic intervention, and that will be done through Professor Simunovic's unit. And then we have our registries, the Fight Retina Blindness Project, where we look to see what happens to these treatments when they're released into general medical use. And we're very advanced in this. We're probably the most advanced unit in the world that tracks the outcomes. And we can identify if people aren't getting, the best results that they could, and we can look for reasons why that might be and make changes in the system. It's often we need to give more injections, for example, for wet macular degeneration. It can be other things too. It may be that we're not getting to it early enough, and we can tell that as well. And in that case, you have to improve the referral pathways. But today we're talking about how we actually come up with the treatments to test in clinical trials, so the laboratory research. And in particular, there's one question that nobody really knows the answer. Why is the macula so prone to disease? So here's a macula with intermediate dry age-related macular degeneration. And all this is just debris under the macula, but outside the blood vessels, like down on the left there, it's completely normal. In fact, it goes way out about 10 times this size, and the disease is just confined to the macula. If we understood why this was, perhaps we'd get better ideas on how to treat it better. The macula is very different from the rest of the retina. It's right in the center of the retina, so it does the straight ahead vision. Most animals don't actually have a retina, so most of the animals we do our experiments in, which is rodents, mice and rats, particularly mice, they don't have maculas. Only humans, primates, and some birds have maculas. So we're limited in the animal studies that we can do. This is a normal looking macula, then obviously it can bleed or it can degenerate. One of the reasons is probably because there's just a lot of cells there. So if you have a look at the cells, this graph here shows the cells, the eccentricity is how far away from the center of the macula it is. So this is the macular part of the central part of the retina, the macula, and then onto the right here, you've got the peripheral retina. There's about twice as many cells in there. They stack up in the macula because they have to do the central vision. And this increased metabolic demand, because these cells here, these photoreceptors, these are the most active cells in the body. And so there's like a furnace in the macula, and it produces a lot of reactive oxygen species. And so this is oxidative stress. So that's probably one of the reasons that the macula is susceptible to degeneration. And you can see here the unique anatomy of the macula. This is the very central vision here. These are the cell bodies of the cells that pick up light, the photoreceptors, and these are their processes. They're inverted the wrong way. But the inner retina, this is all relays out here up to the optic nerve. The inner retina clears in the middle, and so these photoreceptors in the center get the very best view of what you're looking at. It has a unique blood supply. This is an angiogram of the retina, the macula is in the center there. The very central bit doesn't actually get any blood because it doesn't have an inner retina, as I've just shown you. It just has the outer retina, and the blood supply from the outer retina comes from all these blood vessels under here. That's the choroid. But even the inner retina has this is a map of the density of the blood supply. So the more yellow it is, the more blood vessels there are. And there's much more blood flow in the macula, especially on this nasal side near the optic nerve, compared to the peripheral retina there. So that probably is going to increase the amount of oxidative stress and other things that are going on there. Then there can be genetic factors. So MacTel is an interest of ours. It's like macular degeneration, but it has a completely different cause and probably has a completely different treatment. We were the first to find an eye, post-mortem eye with MacTel, and when we cut it up, this is the macula from this first post-mortem eye we found. We stained for Muller cells. So Muller cells are the supporting cells in the retina. You can't have a retina without Muller cells. And they're very important for supporting the photoreceptors. And it looks like in MacTel, all the Muller cells are gone in the center, and that's why the photoreceptors die and you lose vision. And we know that MacTel runs in families, so there must be a genetic factor that's underlying this death of the Muller cells, and that's the research that we're doing at the moment, trying to understand we know what the genes are. They're all to do with serine synthesis. This is an amino acid in the retina, and it's something that's essential for retinal metabolism. And we've found that people with MacTel have low serine levels, and that can cause various issues and complicated pathways, which I won't go into here, but we're trying to get closer to that. Because we understood that better, we may be able to stop this Muller cell degeneration and preserve vision in patients with MacTel. So that's why these are some reasons why we think the macula might get disease, but the important thing is how do we find better treatments for it? Now, this is a complicated diagram, but I'll take you through, and it shows the workflow through what we're doing now in our laboratory with all the new technology that's available. So the first thing that we stress is we don't like to use animal models. We like to use maculars from human people. We get these donations, people who give corneal transplants to the eye bank. We get the maculars. And surprisingly, if you put the retina into a test tube and give it the right culture medium and oxygen, it actually comes back to life for about a week. So we use these maculars, which are cut out, and we put them on a chip and we perfuse them, and we can study them for a week and we can stress them and see how they respond to stress. Then we do these multi-omic analysis both on these maculars, but on disease tissue and normal tissue. So this tells you the genome, the genes, that's the genomics, the RNA or the transcriptomics, that's what comes off the genes, that's the code, and that makes proteins, which is proteomics, and the metabolites are also there. So these are the things that we can study now, and we can study the genes and the transcriptomes and the proteins in a whole piece of tissue and compare that with normal. But even these days, you can sort all these cells. You can get a piece of tissue and make it up into individual cells and sort them out with these lasers. And you might get 60,000 cells out of a single piece of tissue. And each cell, you can do the transcriptomics and the genomics and all these things. And you can also do this omics analysis also spatially. So we can do an omics analysis in the macula and in the peripheral retina and compare the difference between the two there. So you get masses and masses of data. It's literally going through the roof, the amount of data we can get on disease versus normal tissue. And you do these bioinformatic analysis, and you can cluster the cells into all these different cell types. So these will be the rods, these will be the cones, they're the photoreceptors, these will be the Muller cells. And then you give it to the computer, and through artificial intelligence, we know that's going through the roof too. Artificial intelligence, it will tell you what the treatment targets are. So it gets all this information and it looks where all the where they're converging. What are the important sort of choke points and checkpoints in the pathogenesis of the disease? And then it just spits out what the treatment targets are. You never know how it identified what they were, but it will tell you. And it may be a signaling pathway, it might be a cell, it might be a gene. So in this experiment, for example, we've taken 2 eyes and we've punched out a macular and a peripheral retina, and some of them we've exposed to light and some of them we've just got under normal light. And we're going to have a look at how the macula responds and how the peripheral retina responds to this light stress. You do the things I've just talked about, and you cluster the cells. Here's another cluster here. And if you have a look at the clustering of the Muller cells, interestingly, you get 2 very different types of Muller cells. These are the ones in the periphery, and these are the ones in the macula. So they cluster differently. So this actually may be one of the reasons that the macula is different from the peripheral retina, because they have a very different Muller cell population. And interesting, these Muller cells in the peripheral retina, if you expose them to stress, then they will bring up all these stress markers, which are shown on, this is just the gene readouts, the transcriptomes for the peripheral Muller cells who've been stressed here. Compare this to the macula has no stress response genes. So why would that be? You can imagine if you didn't have stress response genes, then you might not be able to cope well with stress. The thing about the stress response in the brain is it actually reduces function for a while. So if you were having like some bleeding or some scarring in the macula, if you try to clear that up, it's going to, your vision will go down if it's in the macula. If it's in the peripheral retina, it doesn't matter so much. But if you're living in the jungle, then you would go blind. You might lose your vision quite significantly temporarily, to the extent that a tiger would eat you and you didn't have babies. So the next person who came along who didn't have stress response genes in their macula might survive very well. And so this is perhaps how evolution has shifted, and we've got maculas that don't have stress response genes. And this is something that's very interesting to us. We're following that up to see if it can translate into treatments. So once you've got the treatment target, you've got to get it to the cell you want in the retina. And there's a lot of barriers to get it into the retina. Even if you inject it into the eye, which we do there's the internal limiting membrane at the top of the retina, and there's other membranes that these things have to pass through to get to the cell you want to. So so there's the barriers. You also want to get to a specific cell type, because if you're going to sort of change the genes, you only want to do that in the cells that need it, and you don't want to do it in the cells that don't need it. We'd like it to carry different forms of cargo, but just different drugs. They might be large molecules, they might be small molecules. but some of the things that we want to introduce these days, especially if you're talking about gene editing, are actually quite large, and we can't carry them with our current sort of carriers, which tend to be AAV viruses. And we'd like things to last for longer too. So we've been developing these lipid nanoparticles where they're basically fat droplets. And by varying the components, they've got all these different components. You can see this, they'll form a ring, a sphere, and then you can put things inside them. So these are the drugs, or in this case could be RNA. We like to use RNA therapeutics, which we get from the University of New South Wales. And just like, this is very similar to what they did with the COVID vaccine. The AstraZeneca COVID vaccine was a lipid nanoparticle encasing mRNA therapeutics. Well, we're doing this now for the retina. And if you vary these compositions, then they have different characteristics, and they will go through different types of membranes and barriers, and they will try to go to different types of cells depending on how you make them. So this is the way we do it. Basically you have a micro mixer, and you feed, this is all tiny stuff, and you feed this stuff in, the lipids are in the ethanol, and there's water on the outside, and it comes in here and it makes these lipid nanoparticles. So then so for example, we want to now see whether these nanoparticles are working. So we'll use this a protein called green fluorescent protein. This is very handy because it glows green, and you can see it if you put it into the tissue. There's no doubt about it that you got it there. So we make up the a gene segment with the gene sequence for this green fluorescent protein, and then we load that into our lipid nanoparticles or fat droplets, and we put them into these macular on a chip that I was talking about. They're human maculars. And then you cut them up under a microscope and see if the GFP got in there, and you can very clearly see that the green fluorescent protein is being expressed here in what we would call this. These are the photoreceptors on the bottom. We'd call this inner nuclear layer. This is the first relay, and this is where the Muller cells live, the supporting cells. So this is getting our GFP into the Muller cells, and these are the cells that we want to be able to treat to prevent MACTEL, and probably important in diabetic retinopathy. We don't know the contribution of the Muller cell to macular degeneration yet, but we're interested in that. So getting the treatment from the lab to the patient, as I've said here, is the drug discovery and development through this very high technologies, biotech stuff that's going through the roof. Just like AI is going through the roof, so is biotechnology. And the amount of information that you can get out of a single cell, and multiply that by 60,000, then you can get you can get the lipids and the metabolites and the genes and the transcripts. There's masses of information, and it's too much for us to comprehend. You have to use artificial intelligence, but we do have this in whatever they spit out, we can test it in these human maculars on a chip, and we can then develop treatments for the clinical phase 1 clinical trials. We're working, as you can see, on the drug delivery, and you can modify these things to maybe last longer. You can certainly get them to pass barriers you want to pay. And if you put little antibodies on them, you can get them to go to the cell that you want, which is often the photoreceptor, can be the Muller cell. And then we test them before clinical trials as much as we can in human tissue. So in human primary cells, so just the cells in culture. We do use some animal models, but we prefer to use the human macula on a chip for the testing. And then our aim is to develop at least 2 new mRNA treatments for macular disease within the next 5 years. And we're actually fairly close with one of them. That could be 2 years. This is the Macular Research Group. Ling Su has been the long-term head of that. And we have some postdoctoral and research fellows. We collaborate with the pharmacy. The RNA Institute is a $500 million initiative set up by the state government, University of New South Wales. They want to get mRNA therapeutics into the clinics, and these are overseas collaborators in metabolomics and bioinformatics, a big lab there. So I think we'll stop on that now, and we'll take questions. So if you want to put some questions in the Q&A, you're welcome to write them in now. I think there was some questions before the meeting on MacTel, where we are with MacTel. So I can tell you that from starting 20 years ago, where we had no idea about this condition, we now have just completed 2 phase 3 clinical trials of the ciliary neurotrophic implant. This is certainly not just us. This is there would be now 50 centers around the world who's part of this collaboration. And this implant, you sew it into the eye, and we've found that that reduces the rate of degeneration of the photoreceptors from anywhere from 20 to 45%. So that could be delaying the loss of vision people would get to by up to 6 months for every 2 years of treatment. There's some more information about that will become available in due course. It has not been approved by the American Food and Drug Administration yet, but it will be going up to them later this year. And hopefully we might see that in Australia next year, perhaps, and hopefully we'll get that reimbursed through the PBS. I thought there'd be lots of questions, but there's none coming through.
Maybe later on.
Maybe later on. You're free to do it. You just got to go to this the chat Q&A, not the chat, the Q&A thing down the bottom there, and you can type your question in there. So if you've got anything that you'd like to ask, perhaps you can put it in now while you're listening to Matt as well.
If anyone's got any problems with the Q&A, somebody just messaged about it. If you can't use it for some reason, just put them in the chat. That's fine.
Regeneration research in MacTel. Yes, look, from A. Bird, that's funny because Alan Bird is the Chief Scientific Officer of the project. Andrew, yes, look, obviously that's the holy grail for everything. It's also the holy grail for advanced age-related macular degeneration. The problem with replacement is that no one's been able to make it work yet. So basically what you need to do is replace the photoreceptors in these retinas. In atrophy, in macular degeneration, the rest of the retina is still there. So that is probably going to be a bit easier. At least to start with, it's just the photoreceptors that are missing, and all the other relays are there, including the Macular cells. In MacTel, the Macular cells are not there. The central retina is really gone. So it's more than just replacing the whole, just the photoreceptors. And even for atrophic macular degeneration, we haven't been successful in replacing the photoreceptors yet, quite frankly. No one's even got close in animal models. Well, they're starting to report 1 or 2% of photoreceptors are getting in. But at the moment, that it's certainly something that's very interested in, but everyone's very interested in. People would love to be able to do it, but it's a lot more challenging than something like a skin graft, where you just put it there and it doesn't have much else to do. Once you put in these photoreceptors, they have to link up with the whole retina, which is only really done during embryogenesis. When we're embryos, it doesn't happen again. And these are adult retinas. So there's many challenges to that. We're working on it very hard, but that's not something that's going to happen quickly. At the moment, the best thing we can do is preserve the photoreceptors and slow their degeneration. We previously weren't able to do that, but with these clinical trials that I've just mentioned, they haven't even been reported yet. This is inside information. It looks like we will be able to significantly slow the photoreceptor degeneration. The eventual human treatment for stem cells, I think I've just been through that. In MacTel, if you put the photoreceptors in there, trouble is their supporting cells aren't there. So in macular degeneration, the supporting cells that are missing are the retinal pigment epithelium. So definitely in macular degeneration, if you do photoreceptor transplants, you'll have to put RPE, retinal pigment epithelium, in there as well. That's not all that difficult, actually. That's not the hardest part, because that goes in a flat layer of cells. and you can grow that on membranes, and you can put it in under a stem cell transplant, that probably would work. But at the moment, Mueller cells don't grow as a flat shell. Well, they do if you grow them in vitro. That's what they have. That's not the way they are anatomically. I guess the thing is that the way you grow Mueller cells in a test tube is the way they look anatomically, just a flat layer. But Mueller cells are not a flat layer at all. Mueller cells are spindly cells that wrap around all the photoreceptors and every cell in the retina the Mueller cell touches. And we think that the MacTel comes about because the Mueller cells die. So if you just put new photoreceptors into a MacTel macula, they won't have the support anyway. So that's the problem. You'll have to put them in with the macula, at least with the Mueller cells. You really need the whole thing. Has the incidence of macular degeneration increased? If so, are there environmental or dietary factors which may play? I'm not aware that the incidence, the actual incidence and the rate that occurs per year is going up any more than anyone expects. The age that which you get it has remained the same. You know, most people, it's very common to have it in your early 80s. Most of my patients are early 80s. Early 80 used to be grand old age. Now it's just a young person. You know, early 80s is like 90 is a new 80. So there's plenty of people in their 90s now. So we are seeing a lot more macular degeneration, but really it's the aging that's occurs in the population over the last 15 years in our lifetimes, which has just been amazing. I'm sure you'll all agree. I don't think that anyone suspects there's anything out there which is like systematic exposure to some sort of toxin, or diets, which is doing that. Macular on chip. Well, basically, you just put the macula. Organ on a chip is just a scientific concept these days. It's growing human tissues in a test tube, not necessarily on a computer chip. And it's not sort of linked up to the computer chip and doing anything neuronal. It's just sitting there, and they're quite sophisticated systems. You have microfluidics to feed the liquids in and drain it all away. And you mount it and you seal it so it's a barrier, so you can put things on one side and things on the other side. And as I say, these maculas come alive, and so you can do these experiments. So as I just showed, we can put in a protein. We use GFP into the solution around it, and that will penetrate into it and get into the macular cells we're interested in. Now, if we did that in an animal, it may or may not get in, but that doesn't mean it gets into a human because they're different, and they don't have maculars anyway. So you wouldn't be knowing if it was penetrating the macula. That's the advantage of the human macula, growing maculars in a test tube, which nobody ever thought you'd be able to do. They thought it died with the person and it was just dead. But no, surprisingly, it comes back to life, and will detect light and do everything for about a week. How do we speculate treatment of new Mueller stem cells? Look, that is a very, very, we're not even there yet. As I've been saying, it is very challenging. It would be very challenging to Mueller cells in there. We're thinking about that. We are seriously thinking about that at the moment. We are seriously thinking about cell therapy in stem cells in MacTel2, but there are many challenges there, and it is quite a way away. Although, you know, I mean, we knew nothing about this disease 20 years ago, and here we've got a treatment for it. That's amazing progress. So with the same focus, maybe it's going to take another 10 or 15 years. Maybe we'll have another thing on that front, but it won't be something that'll happen quickly. All right, so look, that's me from now. I might hand over to Professor Matt Simunovic, who also in the Macular Research Group, and for his presentation.
Thanks, Mark. Thanks for the introduction. This evening I'll be covering emerging treatments for retinal diseases. So it's a pretty expansive topic. The reason I'm covering this or these topics is because I'm often asked about them in clinic, and I thought I'd cover a little bit about what has been done to date and what we're working on in my research group. Just as Mark outlined for the Macular Research Group, my group, the Retinal Disease and Rescue Group, is active in a few domains, including lab research, which I'll be talking about briefly tonight. We're also active in the domain of clinical research. We're particularly interested in assessing visual function in the clinic, both to assess disease itself, but also treatments for disease. More recently, we've been interested in using AI to predict function based upon structural images that we obtain in clinic, and that's in collaboration with researchers at the University of New South Wales and a colleague at the University of Sydney, Andrew Chang. We're also active in clinical trials, in both investigator-initiated trials, looking at different means of delivering drugs and cells to the subretinal space. We've been involved in neuroprotective agent trials, the CNTF implant that Mark mentioned earlier on, as well as an agent that was investigated for use in patients with macula involving retinal detachment. We're also running a gene therapy trial in conjunction with Mark's group, looking at gene therapy for dry macular degeneration. And we're very interested in registries, specifically looking at real-world outcomes for patients undergoing retinal gene therapy, which I'll touch upon a little bit later on. So as I said, I'll cover specifically emerging treatments for retinal and macular diseases, including gene therapies. And this includes both specific gene therapies, which would be most pertinent to inherited forms of retinal and macular disease, and so-called causative gene agnostic approaches, which are particularly relevant for most forms of age-related macular degeneration. I'll talk briefly about electronic retinal implants, concentrating on two devices that received regulatory approval internationally, and I'll cover briefly some of the collaborative work we're doing at present, which is currently in vitro work. And finally, I'll cover cell therapy very briefly. Mark's already touched upon this. So who might these treatments be suitable for? Well, the two principal target populations are people with inherited retinal diseases, including inherited macular disease, as well as patients with age-related macular degeneration. Inherited retinal disease is important because it's the most common cause of blindness registration in developed countries, we believe. That's for people who are children and people of working age. For people 50 or over, macular degeneration is the chief cause of irreversible sight loss, as you all know. Advanced macular degeneration affects close to 1% of the over 50 population in Australia. So how do these emerging treatments work? Well, broadly speaking, gene therapy uses DNA, which is most commonly delivered via a virus, as Mark referred to earlier on, where the DNA is introduced into cells and it acts as an instruction to make proteins that act as a drug. This is the most common approach. Retinal implants use electric currents to stimulate surviving nerve cells within the retina and the macula, and cell therapies work either by delivering factors which promote the survival of cells and their function, or which replenish or replace lost or dead cells. So as I mentioned, broadly speaking, gene therapy is the use of DNA to act as a drug. It's often said that the eye is an ideal target for gene therapy for a few reasons, which are also true for cellular therapy, which I'll be talking about later. So first of all, to get out because it's an organ. Secondly, it has something called immune privilege, whereby it's slightly sheltered from the full force of the immune system. It's a small organ, and this means that only small treatment doses are required, which are easy to produce. And this, combined with the fact that the eye is compartmentalized, limits unwanted side effects for the rest of the body. Finally, it's a paired organ, and this means that if we run an early phase clinical trial, we have an inbuilt control. The main approach for gene therapy to date, as I referred to earlier on, is so-called gene replacement or gene augmentation, where you deliver DNA, it then produces a protein which has the effect of the drug. And the most popular means to date of getting genes into cells for gene therapy is via the adeno-associated virus. When this virus occurs naturally, it doesn't cause any known human disease. And for the purposes of gene therapy, it is inactivated. We know that it works particularly well in tissues that have cells which don't divide and which are mature, such as the retina, where it will induce protein expression for long periods. So it's good for use in the retina. There are other emerging approaches which Mark referred to earlier on. There's a limit, the disadvantages of AAVs to the size of the genome or the genetic information which it can carry. And so it can accommodate many of the genes that cause inherited retinal and macular degeneration, but it can't carry all of them. And the case in point here is ABCA4, which causes Stargardt disease, which is the commonest cause of inherited retinal or inherited macular degeneration, I should say. So the first approved retinal gene therapy, and in fact, the only approved retinal gene therapy, is for a condition called Leber congenital amaurosis. So this affects not only the macula, but the retina in its entirety. It typically causes profound impairment of vision in infancy. It's inherited. It affects between 1 in 33,000 and 1 in 50,000 individuals. 30 different genes have been described as causing Leber congenital amaurosis. About 6% of patients have alterations to a particular gene, RPE65. So RPE65 produces a protein of the same name. It's crucial for the recycling of the derivative of vitamin A used by the eye to detect light. So if you don't produce normal RPE65, you cannot recycle this derivative of vitamin A, and without that, you cannot see properly. So gene therapy in this condition, that is RPE65, inherited retinal degeneration, works by delivering an adeno-associated virus which carries the normal RPE65 gene. This is introduced into retinal cells. They start producing the normal RPE protein, and the recycling of vitamin A commences properly within the eye. How do we deliver gene therapy to the retina? Well, one way in which we can do this is via a micro keyhole operation called vitrectomy. I'll show you a video of this shortly. In the clinical trials of gene therapy for RPE65-related retinal degeneration, the sensitivity of the retina in treated patients improved profoundly, so by up to 10,000-fold. So this is very significant. What did this translate to in terms of visual function? Well, patients experienced meaningful improvements in activities of daily living, and particularly at mobility tasks. In the pivotal or phase 3 trial, on average, patients improved their retinal sensitivity by just over 100-fold. The commercial product is called Luxturna. It was approved for use in Australia in 2020, and I, together with colleagues at the Children's Hospital Westmead, performed the first surgery to deliver Luxturna in November of 2020. I've got a surgical video top right there of one of the cases we've performed. To date, we've treated a total of 12 eyes in Sydney. On average, retinal sensitivity in these patients has improved by close to 100-fold, really mirroring what was found in the phase 3 clinical trials. As touched upon earlier, we're tracking real-world outcomes via a new registry called Fight Inherited Retinal Blindness, which both Professor Gillies and I are involved in. So this slide summarizes the surgical steps in delivering retinal gene therapy. The first step is on the top left, so we need to gain access to the eye. We do this via micro keyhole incisions, and we place ports into the eye, which allow us easy access, and they have one-way valves on them to prevent the reflux of fluid. At the top left there, you can see there's an infusion line which keeps the eye inflated whilst we're performing surgery. The first step of surgery is the bottom left here. We use a small cutting device which also has a vacuum attached to it. So we cut the vitreous, the gel that fills the majority of the eye, and we remove it by sucking it out of the eye using this handpiece, a vitrector handpiece. Once this has been performed, we then perform the injection into the subretinal space. So we do this in 2 steps in Sydney. In the first step, which you can see in the top here in the middle, we inject an inert solution called balanced salt solution into the subretinal space, and we confirm using an OCT, which you can see here, that we're in the correct tissue plane. This means that we don't inadvertently inject too deeply or into the vitreous cavity where we see loss of efficacy. And also potentially a lot of inflammation. Once we've confirmed that we're in the correct space, we then inject the gene therapy solution. We inject a total of 0.3 of a mil. It raises a really large bleb, which is just at the edge of the central macula here, which is seen in the, you can see it's slightly darker here in the central macula, corresponding to the macular pigment. So we perform this injection slowly. Once we've injected the full 0.3 of a milliliter, we then check the peripheral retina. If there are any holes or tears there, we treat them with either a freezing probe or with laser. Finally, we fill the eye with an air bubble and we position patients so that this fluid will gravitate towards the central macula. And that's the operation over. So in the future, it may be possible to treat macular degeneration using gene therapy. I spoke last year about approaches to treat wet macular degeneration, so I won't talk about those now. It may be possible in the future to treat dry macular degeneration using gene therapy to improve cell survival. We heard a little bit about ways of improving cell survival from Professor Gillies, and we're currently involved in one such trial that is currently enrolling at the SafeSide Institute in Sydney Eye Hospital. I also mentioned that I'd talk about causative mechanism agnostic approaches, and one such approach that we're particularly interested in my lab is called optogenetics, which might be suitable for patients with advanced sight loss from dry macular degeneration, as well as inherited forms of macular degeneration. We know that the mechanism of sight loss in these conditions is through loss of the light detecting cells of the retina, the rods and the cones. However, we also know that the cells that normally relay information from the rods and the cones to the parts of the brain involved in vision are often well preserved. The problem is they're not normally light sensitive. So what optogenetics does is it aims to make these relay cells into new light detectors by using gene therapy to introduce new light sensitive proteins to these cells. Our group has been investigating a few different proteins to restore vision via optogenetics in retinal and macular degenerations. These proteins can be from microbes. There are two that we've been using. One is called breaches, the other is called carmine. We can also use human proteins, and we're using one such protein, which is from the normal cones of the eye, which we are delivering to the retinal ganglion cells or the relay cells. On the right here, you can see some optogenetically treated retinas. Mark referred earlier to green fluorescent protein, which we use to tag protein expression in the eye. And you can see that there's robust expression in this retina within the retinal ganglion cells of our optogenetic gene therapy. We published on this recently in Scientific Reports right at the end of last year. We're able to demonstrate restoration of high sensitivity vision, I should say, with breaches in a preclinical model of retinal degeneration. These are some of our more recent results from the lab using a newer generation optogenetic construct called carmine. It may be more sensitive than breaches. On the top right panel, we have responses from a retinal ganglion cell, which normally just demonstrates noisy response like here in response to light. You can see here that it's spiking in response to increasing intensity waveforms. We hope to begin clinical trials of optogenetics in Sydney within the next 3 years. I'm going to move on now briefly to retinal implants. The proof of concept of retinal implants really came in the 1960s with direct stimulation of the visual cortex with electrodes during neurosurgery in awake patients in Cambridge, and also from soon after indirect electrical stimulation of normal eyes and those with retinal degeneration, which demonstrated that electrical activation of the retina produced a visual percept. All retinal implants share similar components, broadly speaking. Firstly, there needs to be a means of capturing an image. This image must then be processed and encoded. This code has to be delivered to a stimulator, which then produces an electric current to stimulate surviving cells via electrodes. There are three key approaches which have been described to retinal implants. First of all, the implant may sit on top of the retina. This is called an epiretinal implant, which you can see up here. These are comparatively easy to insert, but they bypass a lot of the image processing machinery within the retina, which we heard about earlier on. Subretinal implants sit underneath the sensory retina and above the retinal pigment epithelium. They're much harder to insert compared to epiretinal implants, but their advantage is that they utilize a lot of the residual signal processing machinery of the retina. And finally, there are suprachoroidal implants which sit outside of the choroid, which I won't be covering tonight. So two devices have previously gained approval internationally, and I'm going to be talking about both of these. The first to achieve regulatory approval overseas is called the Argus 2 device. It has an image acquisition device which is mounted in a pair of spectacles. This captures the image, which is then transmitted via a visual processing unit, which is worn on the belt to the eye. The device itself within the retina, you can see here the most advanced one, the Argus 2. This is the Argus 1, which in effect was a modified cochlear implant. It has 60 electrodes, gives a similar amount of visual field to a standard Amsler grid. It permitted rudimentary vision, I think it's fair to say. So the best recorded visual acuities are less than the limit for legal blindness in every country. About 80% of the devices lasted through to 5 years in the clinical trials, which is not bad considering the hostile environment of the eye. However, the company no longer supports or makes this device. It's currently pursuing a brain implant called the Orion, which is inserted into the visual cortex. The other implant to achieve regulatory approval was a subretinal implant produced by Retina AG, the Alpha IMS and the Alpha AMS. It doesn't use any spectacles. Everything for image acquisition is within the eye itself. So it harnesses the optics of the eye. It had a 1,520 electrode array. It gave a similar field to the Argus 2. The surgery is way more complex. The power supply itself is housed behind the ear under the skin in a recess which is created or bored into the bone. The Alpha series of implants achieved the best recorded visual acuity to date for any electronic retinal implant, so up to 660, which is the cusp of legal blindness for most countries. This is one such implant under the macula of a patient we treated when I was a fellow in Oxford 9 years ago. And this is a surgical team, 4 of us. It took 8 hours to do the surgery. So it's highly complex surgery. How do patients fare? Well, only around half or just less than half of patients found the implant useful, despite the fact that it appears to be better in terms of visual acuity to the Argus 2. The company that made the implant, Retina AG, folded in 2019. So what are the future directions? Well, several groups internationally are interested in producing thinner implants placed underneath the retina, so subretinal implants which are powered by incident light. We're collaborating with Matt Griffith's group in materials science at University of Sydney, who are developing one such device that has the ability potentially to encode colour vision. This is still in the very early phases of preclinical development. It's in the in vitro stage, which Mark referred to earlier on, and we published on this earlier on in the year in Advanced Materials. So finally, I'm going to touch upon cell therapy. As many of you will know, or all of you will know, pluripotent stem cells have the ability to generate any organ or organ part. They can come from a variety of sources, including embryos, but these days it's also possible to make them from mature or adult cells that are treated with appropriate factors. Many tissues themselves have their own population of stem or stem-like cells. So, for example, we recently found that the adult mid-peripheral retina spontaneously produces progenitor cells, which are seen in this insert here. We found this out serendipitously through storing surgical specimens for the purposes of gene therapy to perform experiments. We found that these explants produce sprouting cells, which appear to be progenitor cells. We published these findings in Retina recently. All of the clinical trials of retinal cellular therapy to date have used RPE cells. The two chief approaches are either to inject a suspension of cells underneath the retina, which you can see here. The problem with this is that it results in a relatively disorganized mass in the subretinal space. Alternatively, and Mark mentioned this earlier on, it's possible to transplant sheets via a cut made in the sensory retina. These sheets replicate better than normal anatomy, but the insertion, it's fair to say, is more complex than simply injecting underneath the retina. The largest trial of injecting RPE cells was conducted by Steve Schwartz's group in California. They injected underneath the neurosensory retina between 50 and 200,000 RPE cells in patients with either dry macular degeneration or Stargardt inherited macular dystrophy. The surgery, which you can see down here, resulted in pigmentation underneath the retina, as well as in some patients, pigmented sheets on top of the retina. They found that all of their patients had either preserved or slightly improved visual acuity. On average, at 1 year, treated eyes improved by 3 lines on a standard letter chart, and that is for both patients with Stargardt disease as well as dry macular degeneration. However, the control eyes also improved over this period of time, possibly through practice effects. More recently, patches of retinal pigment epithelium have been transplanted in clinical trials. Lyndon de Cruz's group a couple of years ago reported on 2 patients who underwent such transplantation. Both of them had large hemorrhages underneath the macula from wet macular degeneration. So these transplants were of patches measuring 6 by 3 millimeters containing 100,000 cells. Both of the patients improved their visual acuity by several lines on a standard letter chart. One of the patients suffered a retinal detachment, which required further surgery. It remains unclear as to whether concurrent surgical removal of both the new vessels, which had caused the wet macular degeneration, as well as the hemorrhage itself, may have accounted for some of the improvements seen in vision. So the funding for this project was pulled by the Wellcome Trust, and it's no longer being pursued. In the future, as Mark mentioned earlier on, it may be possible to transplant photoreceptors, and multiple groups have been working on this, and there are very promising results from animal trials, but it's yet to translate into human clinical trials. I'm going to talk about bogus clinical trials because they've been of interest over the last couple of years. And in particular, there was an infamous case of a clinical trial of retinal cellular therapy conducted in Florida. In this so-called trial, 3 patients with relatively good to poor visual acuity were recruited. Each of them had dry macular degeneration. All 3 patients had fat cells removed from their abdomen, which was then mixed with platelet-rich plasma. This mixture was then pretty much immediately injected into the eye via an intravitreal approach. All 3 patients suffered from complex retinal detachments within 1 week. This is a retinal detachment in one of the patients with a large retinal break here. The retina is all scarred up, presumably from the fat cells that were injected. These retinal detachments, I should say, necessitated multiple surgeries, and all patients ended up legally blind and in some cases not being able to perceive light at all. At present, there are around 200 clinics in North America which offer stem cell therapy. Around 20 of them offer stem cell therapy for retinal diseases, and it appears that Australia isn't immune from this phenomenon. So our research group is working on characterizing fully the progenitor cells, which I referred to earlier, which we've been harvesting from living human donors who undergo retinal detachment repair. We also aim to use these for therapeutic purposes. We're also collaborating with Anai Cordero's group at the Children's Medical Research Institute in Sydney on photoreceptor transplantation. This was recently funded by the MRFF. So in summary, we're seeing that Luxturna gene therapy for RPE65 related retinal degeneration has been approved in Australia, and we've treated 6 patients to date. Our treatment outcomes mirror those seen in the phase 3 clinical trials. in the future, causative gene independent or causative mechanism independent approaches for inherited and age-related forms of macular degeneration may be available. 2 electronic retinal prostheses have been approved previously internationally. Neither of them are currently produced. Multiple groups are working internationally on developing thin film approaches which are powered by light. Stem cell-derived RPE transplantation, broadly speaking, appears to be safe. We're working on living donor human cell therapy as well as photoreceptor transplantation. And finally, bogus stem cell therapies are out there and they may be offered by the unscrupulous. So I should thank my collaborators as well as the various funding bodies who support our research, including the Macular Disease Foundation Australia, the NHMRC, Sydney Eye Hospital Foundation, the Macular Society USA, and the MRFF. Thanks very much for your attention.
Thanks, Matt. That was great. Oh, there's lots of questions. You see.
What happened to the bogus clinical trial doctors? There's scant evidence on this, but my understanding is that they are still practicing. There was a similarly, I don't know how to describe it euphemistically, but similarly dodgy doctor operating in Florida who was pardoned by Donald Trump a couple of years ago. In regards to retinal implants, it seems there's been continued use of funding and financial viability preventing multiple iterations for improvement. How do you think this issue should be tackled? Does it necessitate a different approach? Yeah, look, that's a very interesting point. And, you know, the big problem with any treatment is that you need to not only demonstrate that it's successful in terms of improving function, but you know, governments need to know that they're getting bang for their buck. So there needs to be kind of demonstration of efficacy, normally demonstrated using something called a cost per QALY or quality adjusted life year. And sometimes this can be really, really hard to compute for certain diseases, depending on the outcomes that were chosen in clinical trials. You know, the way in which we assess outcomes has been changing recently. It was touched upon previously by Mark. So nowadays, the FDA, for example, in the USA will accept you know, structural outcomes for certain diseases. And for this reason, for example, pegcetacoplan has been approved for treatment of dry macular degeneration, but it's yet to be demonstrated that this kind of confers a you know, functional benefit. In the future, I mentioned using AI to demonstrate or to predict function, I should say. So one means around this would be to demonstrate using AI that a structural benefit translates to a functional benefit. In the arena of electronic retinal implants, it may be the case that we need to come up with new outcome measures to assess these treatments. So case in point would be retinal gene therapy in the form of Luxturna. So they developed a specific mobility test called the multiple luminance mobility test, in which subjects have to navigate a maze under different light levels to demonstrate efficacy. So that may be an important part in sort of tackling this problem.
Right, thanks, Matt. Look, I think we'll probably tie up now. That's an hour. The last question is, what macular disease will most likely, that can't treat now, have a treatment in 10 years? There's not many macular diseases actually that we can't treat now. It's just getting better treatment. So 95% of it is macular degeneration, diabetic macular edema, retinal vein occlusion, and MAC-TIL. They're the main things that we see. There's a few others, but they're the main ones. That we've discussed macular degeneration today. Look, there will probably be better, there'll be increasingly better VEGF inhibitors, which we'll do, and we'll find out better how to use them. We still don't know how much you should actually inhibit the VEGF, because it seems that some eyes that you don't completely inhibit the VEGF. Sometimes if you leave a bit behind and there's the blood vessel is still leaking a little bit, the abnormal blood vessel that grew in there, that may give better outcomes. So we're likely to incrementally improve our outcomes with time. The breakthrough for macular disease will be the photoreceptor transplantation. I personally don't think they're going to get there with bionic approaches, which is where, because, the arrays they had was a 64 array, the best array they've got. There's a million photoreceptors in the retina, and they're all intricately reconnected. So it's unlikely you're going to get that level of vision unless you use that sort of structure. You may be able to reproduce that, and people are looking at that in a bionic thing, but that's next level for that. I think in terms of the photoreceptor replacement, it's very hard to tell what science will be in 10 years' time. We're always astounded by how far we've come. The VEGF inhibitors were completely unexpected that anyone thought they would work as well as they did. These are very gnarly diseases. They're very, very difficult diseases to treat, but we're definitely making incremental progress. And look, there can be a breakthrough. If there were a breakthrough in how to integrate photoreceptors and Muller cells, it might suddenly be easier than we thought it would be. No one thinks it's easy now. No one's been able to do it. But there might be a few little factors that you find that suddenly make it easier. So if there's very hard to tell. And there's a breakthrough in science every few years. Like there's stem cells was a breakthrough, and actually GFP was a major breakthrough. That got a Nobel Prize. But gene therapy was a breakthrough. These things were not anticipated. So let's just hope to continue to improve the things, and maybe we'll get some breakthroughs along the way. And we'll look forward to speaking to you again next year.